My name is Peyton Bennett, and today I will be talking about the cellular thermal stability assay, or CETSA, for drug targeting. So what we're going to be talking about today, first we're going to talk about what drug targeting is, then we'll talk about what CETSA is and why it is important, and then we will briefly discuss the articles and what they looked at, and then we'll talk about building my seminar. So what is drug targeting? Drug targeting is a method of medication delivery in a way that increases the concentration of medication in certain parts of the body compared to others. And there are two kinds of drug delivery. So there's active targeted drug delivery, which is in relation to medications working with your immune system and the antibodies in your immune system. And then there's passive targeted drug delivery, which is working to enhance the permeability and retention effects. So how long those medications are staying in your body, in your cells, in your tissue, that kind of thing. The current methods they are using to do this is small molecule-based targeting, nucleic acid fragment-based targeting, peptide and antibody-based targeting, and cell-based targeting. I will be focusing on cell-based targeting in my further research. The problem with these methods is that they do not allow for the observation of drug binding. So it's impossible to know how much of the drug is actually binding and effective versus how much isn't binding and is getting degraded. So what is the cellular thermal stability assay, or as I will refer to it, CETSA? It's a type of thermal shift assay, and a thermal shift assay, if you don't know, measures the changes in thermal denaturation temperature and the stability of a protein under varying conditions, such as variations in drug concentration, buffer pH, ionic strength, redox potential, sequence mutations, those kind of things. But what it allows for is a quantitative measurement of stability of individual proteins within the physiological context of a cell. So this assay relies on the principle of ligand binding, results in thermal stabilization, or sometimes destabilization. We'll be talking about in terms of stabilization of a bound protein. So the practical CETSA method measures the amount of soluble protein remaining in a cell following heating at various temperatures in the absence or presence of a ligand. And this is important because it allows you to see that binding activity within tissues and cells. So terms you need to know before we get into these articles. The first thing you need to know is CHK1. It is a checkpoint kinase 1. It's a serine threonine specific protein kinase, and it coordinates the DNA damage response and cell cycle checkpoint response, and regulates a number of other cellular functions, including DNA damage repair, gene transcription, embryo development, and somatic cell viability. The other thing you need to know is V1584111, which is the selective inhibitor of CHK1. So the first thing we will be talking about is Article 1, a high content, high throughput cellular thermal stability assay for measuring drug target engagement in living cells. The aim of this paper was to determine and understand ligand protein interactions at the site of drug action in the cell. Since the CETSA method determines the amount of soluble folded proteins remaining in a cell, they hypothesized that heating cells that grow attached to their wells, the well plates they were using, and determining the changes in the amount of the target protein still correctly folded by high content immunofluorescence imaging might be a useful adaptation of the CETSA method for adherent cells. So basically what they did is they took the CETSA method that already exists and they combined it with immunofluorescent imaging to make it easier to visually see these proteins within the cells and the tissues. And the reason this is important is because a component of small molecule drug discovery is determining and understanding ligand protein interactions at the site of drug action in the cell. For a large number of potential drug targets, classical approaches, so monitoring changes to substrate or product generation, are not viable. And so they developed this novel cellular target engagement assay in adherent living cells using the principle of ligand-induced changes to protein thermal stability coupled with high content single-cell immunofluorescent imaging in an attempt to mitigate some of these potential liabilities. And so what they found for adherent cells, the requirement to heat the cells in suspension at high density is in huge drawback. It not only denatures the protein that they are trying to measure, but it also jumpstarts the process of trypsinization and resuspension. And this can alter the physiological and pharmacological aspects of the cell. Additionally, having to treat cells at high cell densities can result in underestimation of the engagement potency with the target. And it makes comparisons to downstream pharmacological assays a lot more difficult. So it makes it very, very hard for them to determine what is happening in the cell after this occurs. So they called their new method HCIF-CETSA, and it allows for a thorough determination of target engagement and adherent cells based on the principle of altered protein thermal stabilization or destabilization in response to ligand binding. And they found, in relation to the standard CETSA method of heating cells using PBS immersion, that caused cell damage to the CHK1 and required them to use the V158411 inhibitor to prevent that. The new method of covering the cells in PBS and sealing the plate and then heating by immersion in a preheated water bath did not alter the CHK1 nuclear fluorescence, so it made the inhibitor not necessary. This is actually shown in the figure on the left. So those three charts are showing the different temperatures. The blue is at 37 degrees Celsius, the red is at 49 degrees Celsius, and it's showing as the inhibitor increases, the fluorescence of the kinase decreases at those different temperatures, which of course the higher temperature is a lot more drastic because of the denaturation happening. And if you look right below that, it shows the results within their well plates. All right, so the next thing we're talking about is mass spectrometry-based cellular thermal shift assay for target deconvolution and phenotypic drug discovery. So what this paper is doing is comparing the standard CETSA method to the newer method that we discussed in the previous paper, but instead of using immunofluorescence, like they did in the last paper, they are actually using mass spectroscopy. So basically what they found in this paper was that the cellular thermal shift assay is well suited for identifying target engagement between ligands and their protein targets. And combining that with the mass spectroscopy methods, it creates a technique that allows unlabeled target deconvolution in complex samples, such as intact cells and tissues, in addition to cell lysates and other protein suspensions. And the way they did this is they used a melt curve. So what you're looking at on the right is the results of the melt curve and how they found how they got these results. They incubated their samples with a saturated concentration of PBS and then subjected them to a temperature gradient. Then they centrifuged the samples, and the superintendent was subject to tryptic digestion and typically tandem mass tag or TMT labeling to enable the multiplexing of the conditions. And in this case, the conditions were temperature, concentration, and time. And this approach enabled the detection of proteins that are more or less heat stable in the presence of the compound, instead of detecting proteins that vary stability over different heat, over the heat gradient, I guess you could say. So how are we going to make this a seminar? Well, I would like to include more information on drug targeting. I want to look more into the methods, give more background into those, specifically the cellular and the small molecule methods, because those were what I discussed. And then the drawbacks of each of those methods and how they are working to improve those outside of CETSA. I would then like to do further research on the topic itself, so how the assay relates to different proteins and their inhibitors outside of just CHK1, and their specific binding patterns, and then specific ways this will improve drug targeting and how it would be implemented, as well as the fallbacks of using this method. And then as for seminar structure, I would like to better blend, or I would like to blend the papers entirely into the presentation rather than having them as two separate entities like I did today. I want to include more secondary sources and more background information to better support the topic I am discussing. Here are my references, and I thank you for listening to my seminar.